Selective breeding is increasingly recognized as a key component of sustainable production of aquaculture species. However, the rapid development and application of sequencing technology has allowed aquaculture to narrow the gap, leading to the substantial genomic resources for all major aquaculture species. This has opened the door for rapid generation of genome-wide genetic marker database either via the generation and application of SNP arrays or directly via genotyping by sequencing technique. SNP genotyping is the measurement of genetic variation of single nucleotide polymorphisms between number of species. SNP, one of the most common type of genetic variation. SNP marker may be used to measure genetic diversity, population structure, introgression, and conduct qualitative trait locus QTI mapping for trait of agronomic interest, positioning cloning, and genome-wide association studies. This approach, however, are limited by the lack of genome-wide pattern of variation in transcript in most species. In this study was aimed to identify SNP in gene of the whole genome to generate SNP resources for genetic study in tilapia. In short, method of this study is start with downloading 41 tilapia RNA sequence data from NCBI SRA database and apply bioinformatic tool to identify SNP candidate in genes. These data also were retrieved from different individual and different tissue, which provide a valuable resources to discover the SNP in gene for tilapia.
There are 5 main methods in this research. 1. RNA sequence data resources. 2. Discovering SNP with bioinformatic tools. 3. In silico mapping SNP to the tilapia reference genome. 4. DNA sample, primer design and PCR amplification. 5. SNP genotyping and population genetic analysis. Method 1. RNA sequence data resources. Firstly, the 41-run RNA sequence data were obtained through NCBI SRA database, which generated by the Broad Institute USA, Southern University China, and University of Maryland USA respectively. Then, the detailed run description were placed in additional file 1. The tilapia transcriptome were downloaded from Ensemble database. After that, the 9 tilapia reference genome and genome annotation GFF3 file were downloaded from the website. Method 2. Discovering SNP with bioinformatic tools. Firstly, the 41 RNA sequence database were downloaded from NCBI database. The 9 tilapia transcriptome were downloaded from Ensemble database. Then, the low quality rates to the reference transcriptome were filtering using Bauti 2. PCR duplicates were removed and realigned by Picard tools and genome analysis TK. Next, coordinated by SAM tools and SNP calling by VARSCAN. Furthermore, the 9 tilapia reference genome were downloaded from Broad Institute. The SNP in transcript were mapping to the tilapia reference genome by GMAF. Is functional analysis using SNP-EFF. Then, confirmation of SNP and their application in population genetic analysis. Method 3. In silico mapping SNP to the tilapia reference genome. Firstly, the 9 tilapia reference genome and genome annotation GFF3 file were organized in LGS. Then, the transcript sequences with identified SNP were mapping to the 9 tilapia reference genome by using genomic mapping and alignment program GMAP to obtain the precise genomic position of SNP. After that, the genomic position information were derived for SNP data. Last but not least, the functional analysis of SNP with assigned genomic position with the genome GFF3 annotation file were performed by using the generic variant annotation and effect prediction toolbox SNP-EFF.
The next step is DNA sample, primer design, and PCR amplification. Firstly, leaf tilapia, Nile tilapia, red tilapia, and Mozambique tilapia were selected from the 4 population. Then, genome DNA was extracted from fin clip using the DNeasy Black and tissue kit according to manufacturer instruction. Then, the isolate DNA will keep at -20 degrees Celsius until use. Then, 1 kilobyte of genomic DNA sequence contain candidate SNP were retrieved from the 9 tilapia genome sequence. 1 primer pair for each candidate was designed by the program Primer Select like DNA Star, Wilmington, and DE. Then, primer was selected to perform PCR under the annealing temperature of 55 to 60 degrees Celsius. Then, the primer designed at 3 and 5 inch and were at least 100 BP line flanking the SNP of interest. A master reaction mix contains 0.2 microliter of forward primer and reverse primer and 10 mg of gDNA template for the PCR reaction with 25 microliter of total volume. Then run the thermocycler. Next, PCR product for each primer pair, giving one specific pen by using 2% of agarose gel with ethidium bromide. The result which were the pen were observed under UV light. Then the sequencing PCR was carried out using either forward primer or reverse primer with a terminator sequencing kit. And lastly, the PCR product was sequenced with 3730SL then analyzer. The last step is SNP genotyping and population genetic analysis. In this step, SNP genotype was analyzed by using software sequencer 4.9, such as GeneCode and NABO. Meanwhile, the genetic variation conducted using software PopGene. Unweighted pair group method with arithmetic mean or UPGMA phylogenetic tree among 4 populations were inferred based on its genetic distance by using software Megaform. Then the genetic structure of each population was subdivided using model-based approach in structure software.
Okay, I will continue about the result. From the table 1 show the summarize of genetic variation statistic in the 4 different tilapia population, which is gift tilapia, Nile tilapia, red tilapia, and the last one is Mozambique tilapia. Okay, 8 individuals in every tilapia population were selected to get the percentage of genetic variation. Okay, for the gift tilapia have 69% of genetic variation, while Nile tilapia have 52% and red tilapia have 77%, and the last one is Mozambique tilapia is 12% of genetic variations. Okay, from the table we can conclude that red tilapia is more heterogeneous compared to the other 3 population because the red tilapia have the highest number of percentage, which is 77% of genetic variations. Okay, from the data, why the Mozambique tilapia is lower informative marker than Nile tilapia? This is because SNPs originally developed from the Nile tilapia, and maybe Mozambique tilapia have the lowest number of NE in their populations. Okay, that's all for the result, and we continue about the discussion.
Based on the experiment, they will use SNP data, which is to identify the variation of genetic among the 4 types of different tilapia. First is red tilapia, shows the highest amount of percentage in variation of genetic, which is about 77%, and also followed by the gift tilapia, which is about 69%, and lastly is Nile tilapia and Mozambique tilapia, which is about 56% into 15% of genetic variation. For example, red tilapia shows the highest amount of percentage polymorphic SNP and loci, and also heterozygosity, which is about 69.1%. And lastly is Mozambique tilapia, which shows the lowest percentage of SNP polymorphic, loci, and heterozygosity, which is about 26.6%. Spatiotemporal genetic structure is used to reduce the Nile tilapia species. It can be used in a morphological trait, microsatellite loci, and also in a molecular marker. By use the UPGMA of the phylogenetic tree is used to identify the NEIS genetic distance. First is we need to construct the phylogenetic tree. Second is we split the fish into a different group. For example, for the red tilapia, we will mix into a different types of subskelette that have a different mixture of the resources. Next is Mozambique species. Mozambique species were clustered into a ones of subskelette that contain 100 bootstraps of value. For the software structure, the gift tilapia and Nile tilapia shows the different amount of the value than the red tilapia and Mozambique tilapia, which is K equal 2. This is because of a Mozambique tilapia is actually belong to the clade of K2 into K5, and for the gift tilapia and Nile tilapia, it shows clade K is more than 3. For the previous MTD and study analysis, both of Fiji gift strain and also one fish train center is used to reveal a new species of a fish. For example, Oreochromis sp is actually two integration of Oreochromis Mozambique and Oreochromis aureus that change into a gift strain. Next is red tilapia, is about combination of Oreochromis Mozambique, Nile tilapia, and the other tilapia that have more than 3 amount of K in a clade. And lastly is Taiwanese red tilapia, which is crossbreeding of Mozambique female tilapia and also a normal male tilapia. Finally, since the experiment will use LG23 chromosome, it shows that genetic across 4 types of population of tilapia is actually not be able to be used and choose into a wide genome of tilapia. In conclusion, by mining the publicly available RNA sequencing data of the 9 tilapia has resulting a valuable genetic resource consisting of 17,699 gene-associated SNPs for the 9 tilapia. These SNPs were mapped onto the whole genome of 9 tilapia using in silico mapping. Genotyping of a SNP set that selected from LG23 in 32 individuals from 4 tilapia population verified the polymorphism for 93% of the SNPs. Population genetic analysis using SNPs disclosed an admixture in the red tilapia. Since genotyping a large number of SNPs individually by single sequencing is laborious and costly, a SNPs genotyping system such as oligonucleotide SNPs array that was able to economically genotype large number of SNPs in large sample set is required for tilapia in future. The availability of the large number of gene-associated SNPs will facilitate the identification of candidate genes underlying QTL regions, controlling economic traits, as well as a variable resource for genome-wide. Analysis of SNPs has emerged as a genotyping technology with wide and significant application in aquaculture. SNPs can be used for construction of high-density and high-resolution genetic linkage maps, finding QTL for useful traits like growth, body weight, morphological characters, identification of progeny in selection, and chromosome manipulation experiment, as well as assessment of genomic selective in aquaculture. SNPs are used for identification of region-selective sweeps in the genome, which are related to useful traits for aquaculture. SNPs are useful also to identify genes, sequence, and nucleotide, directly influencing phenotype variation, thus opening the possibility of marker-assist selection for desirable characters or traits in culture. If a sufficiently large number of SNPs is discovered and used, mutation gene associated with a trait can be identified. The identified genotypes can be used in marker-assist selection. A large number of SNPs used in a study is feasible for analysis of polygenic traits and facilitate the finding of genotype useful for aquaculture.
In summary, analysis of SNP has emerged a genotyping technology with wide and significant applications in aquaculture. SNP are used for identification of region-selective sweeps, which is containing genes in the genome which are related to useful traits for aquaculture. Single nucleotide polymorphism are important DNA markers for genetic studies. SNP are useful also to identify genes, sequence, and nucleotide directly influencing phenotypic variations, thus opening the possibility of marker-assisted selection for desirable characters or traits in culture. SNP can be used for construction of high-density and high-resolution genetic linkage maps, finding QTL, which is quantitative trait loci for useful traits like growth, body weight, morphological characters, thermal and low oxygen tolerance, resistant to stress and disease, mapping sex determination loci, and identification of progeny in selection and chromosome manipulation experiments and assessment of genetic selection in aquaculture. However, the number of SNP is limited in importance of aquaculture species. The studies has identified 23,535 high-quality SNP in genes of 9 tilapia, Oreochromis niloticus, by analyzing the available RNA sequence. 17,699 SNP were mapped onto 22 linkage groups using in silico mapping. The number of mapped SNP ranged from 332 of linkage group 2 to 1,148 on linkage group 16 to 21 with an average number of 805 SNP per LG. The mapped SNP were located in 7,146 genes. This SNP resource will be useful in monitoring the genetic diversity and mapping QTL for genetic improvement of tilapia species. The relationship among individuals from 4 populations, which are GIF, Oreochromis niloticus, red tilapia, and Oreochromis mozambicus, using 101 SNP from LG23, 94 SNP were polymorphic in the tested population. Red tilapia, which is 77%, has the highest number of informative markers than GIF, which is 69%. 9 tilapia, 56%, and Mozambique tilapia is 12%. Significant population structure was detected through phylogenetic and population structure analysis. The red tilapia individuals were closely related to Mozambique tilapia, but were split into several subclades, confirming that red tilapia is a hybrid admixture to different resources of tilapias.